So today we'll be starting off with cell signaling. I said in the previous video that this is going to be our next topic. It is a continuation of where we left off last time. We discussed this very briefly in the functions of glycoproteins and glycolipids, where we had said that cell receptors for hormones and neurotransmitters in the cell signaling region, right? So we've learned that there's two main control systems, which is the nervous system and the hormonal system. That is what the basis of cell signaling are built upon. So first off, we'll start off with what sensitivity means. Now, sensitivity is the ability to respond to a stimulus. What a stimulus is a change. It can be any kind of a change. It can be an external change or an internal change. Now, for example, if someone touches a hot iron placed anywhere, so for example, the first thing that he'll do is he'll withdraw his hand as soon as possible. That's a reflex action. Now, how it is done is through the nerve impulse being transmitted into the brain to help you respond to the specific stimulus, which stimulus is feeling heat, right? So this is how sensitivity works. So for example, at first there is a receptor and a regulator. Now, regulator is always the nervous or the hormonal system. These are the two systems that are largely specialized. So the receptor gets a stimulus, detects a stimulus, and through the regulator gives signals, and the effector in the end, which is usually a muscle or gland, is able to respond to it. So the final response that you get is that you move your hand, right? So that is the specific response that you're looking for, which is helped to you through cell signaling, through the receptor, the regulator, and the effector. Now, what the definition of cell signaling exactly is the transferring of messages, be it electrical or chemical. Electrical, if it says through neurotransmitters, for example, electrical is through neurons, neurotransmitters, and chemical is through blood or hormones, right? Now, for example, transferring of message from one body part to the other is known as cell signaling, or you can also say the transmission of a message from one part of the body to other specific cells of the body is known as cell signaling. And it can be in two forms, as I mentioned, electrical and chemical. Electrical is through neurons and chemical is in the blood, right? So both of these transmissions, they include or they involve a release of any substance, right? A release of a substance which is received by a receptor, and then that receptor on the target cell receives, for example, a specific instruction or a specific response to be generated, and that specific response is all we're looking for, right? That is what cell signaling is. Now, there's three mode of action of hormones, which is a big part of our cell membrane and transport. So the first thing is direct action on genes. So some hormones, now, because they can directly diffuse, these hormones can directly diffuse through the phospholipid bilayer. So this shows that these are non-polar because the phospholipid bilayer is also non-polar, right? So because of its non-polar nature, it can diffuse directly through the phospholipid bilayer. Now, specific receptors are present in cytoplasm or, for example, nucleus, which help it to diffuse easily. So these hormones can directly diffuse through the phospholipid bilayer, as it mentions right here. and are received by the specific receptors in the cytoplasm or in the nucleus and activate a gene to make specific enzymes for a specific response. So what how this is done is, like, for example, if there is an if there is a specific receptor in a cytoplasm that activates a gene, and that gene further on makes an enzyme, and that enzyme helps to create a specific response to that specific hormones specific response, right? Now what we have to know is that generally most hormones are proteins in nature. They can either be proteins or they can even be short polypeptide chains, which we also call as peptone, right? So peptones or proteins in nature, thus proteins are generally polar in nature. So as they're also polar, and they're sometimes even large sized molecules, so they can't diffuse directly, right? So thus other means are required. Now there are very few of these kinds, the ones that have direct action on genes. They're usually the steroid hormones, which are the non-polar hormones. So most of them are these testosterone, estrogen, and progesterone, and all these three are sex hormones, right? I'll just mention it right here. Right, so sex hormones, all three of these, testosterone, estrogens, and progesterones. So the next case is direct action on cell membrane. These do not have to cross the bilayer as they are acting on the cell membrane, as I mentioned. So for example, these type of hormones act on the cell membrane and are received by specific receptors, which I'll show as we proceed. And these specific receptors can even act as membrane bound enzymes. So as a result, those enzymes can be released inside the cell, which further on stimulate a final response, right? So for example, I'll give you an example. The glucose level, if it increases in the blood, right, in the blood, glucose level increases. So the normal thing that happens, the usual thing is insulin is released into the blood vessels as well. And because, like right here, glucose is a lot outside in the blood, and as its levels have increased, so this specific receptor is attached to two enzymes, and insulin right here, it comes and attaches on this specific receptor. This further on stimulates a response which opens these carrier proteins that we've learned, which are specific to glucose, to be absorbed into the cell, right? And what the cell does is it transforms this glucose into a storage form of glycogen for later use. So the action of insulin right here is to open the glucose carriers that are present specific to glucose, so that the excessive glucose can come in and taken in to be stored as glycogen. So this is what the direct action cell membrane method is, and these are the types, these examples of hormones that travel this way, ADH and insulin. Now there's one last method, which is a second messenger system, and this is the way most of the hormones travel, or as they're water soluble. Most of the hormones are water soluble and cannot diffuse directly through the cell membrane. So this is how they do most of the time. These hormones act through a second messenger system, as we've mentioned. So they're received by a specific receptor. So this is a hormone received by a specific receptor, and this receptor on the membrane usually results in a change of shape, right? Change of shape, and this further usually activates another G protein, and this G protein activates the release of another enzyme from the cell membrane, thus acting as a second messenger system, right? So I'll repeat it once again. A hormone is received by a specific receptor. That receptor undergoes a change in shape. Then it usually activates a G protein which is associated right next to it, a G protein, and then that G protein activates the release of another enzyme that from the cell membrane, right? So this enzyme activates a further more enzymes. For example, one enzyme activates a higher amount of a second enzyme, and then a higher amount, and then a higher amount, so it amplifies it. That's why it's called amplification. This amplification is done so that a higher proportion of the final enzyme is amplified and released, so the signal in the end, the signal that is received is amplified, right? And that amplification is done through this entire system where where it's received the G protein, then activates another enzyme, and that enzyme keeps on activating a higher enzyme, and then a higher enzyme, and then a higher enzyme for a greater amplification of the signal, thus named as a signaling cascade. Now there are two types of enzymes that we majorly know that work this way. One of them is glucagon, and the other one that we've also studied in O level is adrenaline. Right? These two work as a second messenger system. So the last thing we're going to do right now is how the opening of the ion gated channel is usually done through the mode of action of neurotransmitters. So for example, this is a neuron, a presynaptic neuron, and a neurotransmitter is released which is called acetyl. Right? Now this is the cell membrane, and acetyl is received right here in a specific receptor, and then that helps it to open this gated channel right here and help the sodium ion travel inside. So the specific neurotransmitter, for example acetyl, has a specific receptor, and that specific receptor helps to open the specific gated channel right here. So we'll just revise what we've done entirely today. So for example, we started off with cell signaling and how and what sensitivity is. Then we went on to describe what cell signaling is, and then what cell signaling is, and the two types of forms in which messages are transferred. Then we learned the mode of action of hormones. The first action was direct action on genes, where some hormones could directly diffuse through the phospholipid bilayer because of specific receptors in the cytoplasm, and this would help activate a gene to make a specific enzyme inside the cell for a specific response. However, most of the hormones are proteins in nature or peptones. Peptones are small polypeptide chains. So as a result, they're polar and long molecules as well, and big molecules rather, big, so they're not easily diffusible inside, so other means are required. The other way is direct action on cell membrane, in which it doesn't have to cross the bilayer. So for example, insulin is released, it comes inside, it activates an enzyme, it helps the carriers for a specific glucose, so carriers specific only to glucose to open up. The glucose comes inside and it is transformed into glycogen, and then transformed back when the need arises for glucose to be used. So we've done the examples. For example, direct action on gene had steroid hormones, most of them were sex hormones. Direct action on cell membrane had ADH and insulin, and then there was the third one, the second messenger system, where a specific receptor received a hormone and then it changes shape. It activated a G protein. That G protein activated another enzyme from the cell membrane, and then a signal is amplified through the signaling cascade, where one enzyme activates a higher amount of the second one, and then that activates a higher amount of the other one, and then for a third response. So as a result, the signal is amplified by the end, which is required to turn a substrate into a product. The two main examples are glucagon and adrenaline, both of which are used in the liver. The last thing we did was opening of ion gated channel through the mode of action of neurotransmitters. So for example, a neurotransmitter acetyl is released from the neuron. It comes onto the cell membrane and the specific receptor takes it, and this helps to open the gated channel through its means, and the specific sodium ion travels inside.